To meet. I would love to meet all of you in person, but unfortunately, because of the misunderstanding, this has happened. Basically, we should have yellow fever vaccination if we visit the some Latin American countries. So, but unfortunately, none of although I work from Singapore General Hospital and it was through approved agent, we were never informed about this. In any case, that's how the life goes. You need to be prepared for everything in eventuality. And I thank Professor Seeram who has been, telling me, you always tell that, you have to have a backup plan and then the perfect, master executor of any kind of, strategies. Yeah. So with that, we can begin the class or we'll wait for a few more minutes. Do you expect some of your colleagues would like to come or what's the?
You can go ahead. Yeah.
Okay. 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 So basically, this is the second part of the drug delivery that I have taken for this particular academic year. Just to recap, in my first lecture, it was done on October 4th, I think, and we have come across October 1st, I think. We have come across 5 different classes of therapeutics, particularly small molecules, peptide proteins, antibodies, nucleic acids, and cells. And in my first lecture, I covered exclusively on small molecules and what are the different methods of delivering them and the principles behind that. And also gave you some commercially successful strategies that employ these small molecules for a controlled drug delivery. And as I said, the ultimate aim of drug delivery the ultimate aim of drug delivery system is to maximize the therapeutic efficacy and at the same time minimize the toxicity. So if you recall the pharmacokinetic curve after oral absorption, there is a minimum effective concentration and minimum toxicity concentration. So ultimately the drug delivery system has to maintain the the concentration of the API within this window. And I also included different routes of drug delivery, topical, oral, parenteral, otic, nasal, etc. And basically in previous lecture, I covered mostly on the oral, and we are going to see some of parenteral method routes of delivery in this lecture. Then I introduced the concept of pharmacokinetics and the importance of PK parameters, such as Cmax, that is a concentration that reaches the maximum time point, and then the Tmax, area under the curve, and minimum effective concentration and minimum toxicity concentration. So all these are quite important parameters that would help in the device or in the design of appropriate drug delivery system. And I've also come across, and detailed about the different mechanisms of release, diffusion control, dissolution control, and the most successful osmotic control drug delivery systems. Well, these are all quite useful for small molecules. And in this lecture, what I'm going to do is that what are the ways and how the polymers and how the polymer structures can be manipulated to deliver a much more challenging therapeutics such as peptides, antibodies, nucleic acids, etc. Some of the learning objectives of today's lectures are there, and what are the challenges and strategies in the delivery of biopharmaceuticals? And it's been over the years, you could have seen, particularly after the COVID-19 pandemic, you would see there is quite dramatic increase in the delivery of biopharmaceuticals, such as proteins and peptides or antibodies or RNA nucleic acids for to target or to overcome any kind of diseases. So it's definitely there's a huge potential to enter into opportunities for engineers like you in this area. And then what are the polymer chemistry approaches for the delivery of pharmaceuticals? Why we use certain specific polymers to make an appropriate, formulations, and what is their functional role in delivering in these large macromolecular drugs. And third and finally, I'll give you the principles behind the COVID-19 mRNA vaccination. So with these objectives in mind, let's go through one by one. So basically, what are biopharmaceuticals? These are basically derived from biological source, and they could be produced by by bacteria or mammalian cell system, and or they can be produced by eukaryotic or prokaryotic cells, and also they include proteins and peptides, nucleic acids, carbohydrates, and transplanted stem cells and tissues. So all are come under biopharmaceuticals. And when you compare them with the small molecules, which are of few hundreds of Daltons in molecular weight, the peptides and proteins are much more larger, ranging from more than 1,000 to several millions of Daltons in size. And how are they delivered? In general, they are all delivered through parenteral route, that is through by syringe and needle. They cannot be administered orally, largely because they either denatured or degraded by the proteases present in the stomach and also the acidic environment in the stomach. Therefore, the biopharmaceutical drugs are compared to small molecules are less stable. They're than small molecular weight drugs. And also, there's always a storage stability issues associated with these biopharmaceuticals. So they have to be administered as quickly as possible from the production source. Now, what are the key challenges? We have seen for the small molecule, there's always an epithelial barrier that prevent the passage of the drug. And then there's a limit of 600 Daltons. Anything above 600 Dalton cannot pass through this epithelial barrier. Therefore, there have to be, some way of administering them to the host tissue. And and also their therapeutic range is quite variable, depending on how they interact with the target site. It vary from nanogram per kg to milligram per kg. Accordingly, the dosage has to be optimized. And also, finally, the one has to devise a patient-friendly administration routes. Usually, such drugs, large macromolecules, are administered for either old elderly patients or very young patients to overcome the disease. So therefore, it has to be the whatever the administration rules, it has to be patient friendly. Let me begin with the topic of proteins, peptides, and antibodies. I included antibodies also in this because it's also the major constituents of antibodies or the peptide bond. And as you can see, the proteins and peptides composed of individual amino acids linked by the amide bond, as indicated in the circle. You can see the peptide bond is there, and then any of the 20 amino acid residues that can be incorporated to give an appropriate uh, size and varying from a few thousand Dalton to more than 150,000 Daltons. Antibodies, for example, they have a molecular weight of 15,000 Daltons and vary to all the way up to 70 or 80,000 Daltons. So really, there's a big variation in size of the protein or peptide-based drugs. And these such large molecules always come, in many cases, composed of charged amino acids such as aspartic acid, glutamic acid, which are negatively charged, or they may also contain a positively charged amino acids such as arginine and lysine. In addition, they also carry these aromatic amino acid residues such as phenylalanine, tyrosine, tryptophan. Now, I want a response from you. If you want to probe the stability of a peptide or protein, which amino acid do you think is quite useful for assessing their stability? Make a couple of minutes guess and then respond to my question. I'll be very happy if you respond. Those who are online can also do this. There are 20 amino acids, right? So which one do you think is a very useful probe to monitor stability of a peptide or a protein active pharmaceutical ingredient? Anyone? I'll wait for another 30 seconds. Okay, no answer from any of you. So basically, tryptophan is a useful amino acid to probe the stability of any peptide or proteins, largely because it is a heterocyclic aromatic ring containing the indole ring. This indole ring, it has the emission wavelength of vary from 310 to 360 nanometers. So when you excite them with a wavelength of 280 to 200 nanometers, and they have a characteristic emission pattern, if the tryptophan is an hydrophobic environment, if that is because it has a proper fold and buried inside the protein structure, it would have an emission around 308 nanometers. If it is solvent exposed, that is, it is the solvent can readily access this residue, then you would have an emission at 352 nanometers. So basically, the structure of the protein or peptide, if one monitor the tryptophan emission pattern, one would be able to guess what kind of a secondary structure, whether it's a folded or unfolded structure, and that can be useful to monitor the stability of the proteins and peptides. So we talk about proteins, peptides, and antibodies, and these, as I said, the number of approved peptide-based drugs has dramatically increased over the last 20 years. As you can see, the value is almost doubled from when compared to 1980s and 90s. And they usually contains 22 to 50 for peptides and more than 50 for the proteins. And they're considered to be, these are considered as the second generation antibiotics, and they occupy substantially 20% of the total markets. And in addition to that, the peptides and proteins have very high specific targets, and they highly selective towards a particular target. And they also differ very widely in terms of the size, and then the diversity of secondary or tertiary structures. And also, it's very important to maintain the tertiary structures of the proteins and peptide-based API. Otherwise, the properties would vary. And recent studies have shown that around 80 peptide or protein-based drugs are available on the global market, and more than 150 of them are in the clinical development, and several of them undergo preclinical and clinical evaluation. The targeted disease include as vary from a metabolic disorder, the diseases such as diabetic, and all the way up to vaccines as well as the cancer-based therapy. So the application also quite vary significantly depending on the nature of the amino acid or constituent amino acid residues. Now, what are the important differences between the proteins and small molecules? Why we need to have a different strategy for proteins and peptides compared to small molecules? And this is basically a diagram showing a cartoon showing how they differ in that size. And usually the small molecules are less than 600 Daltons in size, whereas proteins and peptide-based drugs vary from 1,000 to all the way to 50 or 60,000, sometimes even 80,000 Daltons in size. So there's a massive variation in their size. Therefore, the molecules or small molecules have very few reactive sites. On the other hand, the proteins and peptides have many reactive sites as it has numerous one of the 120 amino acid residues. In addition to that, there are few ionizable groups in a small molecule. For example, there will be some of the drugs may have one or two carboxylic groups or amino groups. On the other hand, a protein or peptide have many ionizable groups. There are, as I said, there are proteins and peptides which carry a number of ionizable amino acid residues such as aspartic acid or glutamic acid, which are negatively charged, or they may also carry amino groups or guanidine like arginine. So these groups have different ionizable characteristics. And in addition to that, histidine is also another one, which is highly sensitive to pH. So therefore, in a protein molecules, it has a numerous ionizable groups compared to few ionizable groups in a small molecules, which completely change their physicochemical properties. In general, for a small molecule, the buffer action is general. Basically, there won't be much change in their pH. And on the other hand, the small molecule or peptide-based drugs, the buffer action is unique, depending on the ionization PI, that is the ionization constant of the total protein molecule, depend on whether whether the consumed amino acids are acidic or basic. The PI value varies with their total constituent, whether they are negatively charged or they carry more number of um, positively charged amino acids. Small molecules do not have a higher order structure. Their structures can be easily determined by the NMR. And on the other hand, the proteins or peptide-based drugs have secondary, tertiary, and quaternary structure, which make their structure much more complicated. So you need to have an appropriate structure in order to maintain the activity. And usually when we, small molecules behave like a salt solution. They do not um, have any light scattering properties. They behave quite nicely, just like a sodium chloride in solution form. On the other hand, the proteins and peptides form a viscous solution depending on the concentration. And they also form a colloidal solution, which has unique light scattering as well as light scattering and then the rheological properties. In general, the temperature effect is continuous. There's a sharp melting transition for small molecules, whereas for proteins and peptide, they can be discontinuous, meaning that there's a range of temperature over which they melt. In addition to that, one of the major drawback of protein and peptide-based drugs is that, they promote, the microbial growth, which is, so for example, if you're using a parenteral injection, this is an important aspect. The injected formulation should not contain any microbial growth. Therefore, the formulation has to be appropriately prepared in a preservative that prevents the microbial growth. Now, having said that, all the proteins and peptides are composed of amide bonds. What are the important stability problems and how do we address them? This is basically a formulation consideration before when doing that. And the one of first problem is that hydrolysis or deamidation. As I said, when we dissolve a protein or peptide, the pI, depending on the pI, the charge also varies. And how I mentioned that since water is present in large excess, and then the conditions could dramatically change depending on the pH. The, these drugs, these API, the active pharmaceutical ingredients, are prone to undergo hydrolysis or deamidation of the asparagine or glutamine residues. So how do we address them? By having a pH control, that is a buffers of low ionic strength, and also buffers which has less variation, temperature variation. So by using these buffers, one would be able to overcome the hydrolysis or deamidation problems. And many of these antibodies also contain disulfide bonds and also tryptophan and also methionine. And these three important amino acids are prone to oxidation by just aerial oxidation. Therefore, it's important to protect them. Once their oxidation occurs, the structure of the peptide or protein completely vary, and which eventually affect their target properties. So it's always the formulation also, therefore, it's good to add antioxidants, chelating agents, or oxygen-free processing and packaging conditions. So usually the many of these formulations do contain antioxidants or chelating agent, which prevent the aerial oxidation of peptide-based drugs. As the pH is lowered, it's all but as the pH is more than 8, some of the amino acids such as serine and threonine undergo a reaction called the beta elimination reaction, which completely alter the secondary structure and of the protein. So eventually, the best way to do is that for such proteins and peptides, it's good to maintain low pH, and chelating agents would overcome the beta elimination reaction. And another aspect of stability problem is the racemization, the cis-trans isomerization of the peptide bond. Usually that can be arrested by appropriate choice of the buffer. And those antibodies which contains numerous disulfide bonds, they can be mitigated by adding a thiol scavengers, which prevent this. Once the disulfide exchange reaction takes place, then the fold of the protein completely changes. So to avoid that, usually the formulations, and particularly those with the antibodies, have thiol scavengers. And all these biopharmaceuticals are manufactured or in the powder form. If you want to, in the powder inject, either they are in the solution or manufactured as a powder form. And they use the process called the freeze drying. And during the freeze drying, the undergoes possible of denaturation. To avoid that, they always add cryoprotectants or lyoprotectants, which prevent the misfolding of the protein. And aggregation and precipitation are the complex problems associated with the solution form of the peptide or protein-based drugs. Usually, they can be mitigated by pH control or surface active agents or minimize the mechanical stress. All this can be used to minimize the aggregation or precipitation of the proteins. One unique problem associated in particular with the insulin is the absorption to the surfaces. And so therefore, to avoid that, to avoid these one, either the surfactants are added or albumin is the tubing, for example, those who undergo parenteral injection of insulin, the surface is coated with the albumin or surfactants to prevent any surface absorption so that the dosage is maintained throughout the injection period. So there are, in principle, where one can do intravenous injections. The advantage is it's highly beneficial and 100% bioavailability and reproducibility. Unfortunately, it's very painful injections. And actually, they doesn't depend. There's no barrier. Basically, the moment when we do the intravenous injection, it basically reaches the tissue without any However, and it is also useful to use subcutaneous. Many actually APIs and or administered through subcutaneously. And usually, for example, insulin is a good example of self-administered or administered through by the patients or by the specialized nurse. Um, so these basically avoid the first pass metabolism, and unfortunately, though it's a painful injection, uh, some of the barrier properties of the drawback of subcutaneous injection is there. The extracellular matrix is present in the thing, which would take away some of the dosage forms. Eventually, one has to use more dosages than one would easily expected. And also, there's a limited space of the subcutaneous tissue, and which minimizes the injectable volume. So beyond which the drugs cannot be reach the active sites. And numerous proteins and antibodies have been used by subcutaneous in those who are not, to avoid the intravenous injection. Subcutaneous is the option. The other options are inhalation. Again, inhalation is through the nasal route, and then the advantage is that they have the alveoli has a large surface area, and it's essentially a non-invasive procedure. One would use a small device to really deliver the drug. And however, a lot of variations in there, and along the airway pathway, there are a lot of mucus as well as the epithelial cells, which basically take away or consume many of the drugs, eventually decreasing that concentration. The other alternative route is the buccal, and it's again the oral mucosal surface. It's again a non-invasive procedure, and however, the absorption is quite limited because of the low surface area, and also the salivary proteases may sometimes degrade some of the active pharmaceutical ingredients. But nevertheless, by making appropriate formulations, one will be able to make a successful drug delivery system by using one of these routes. So, in summary, what I'm going to say is that the some of the challenges associated with the biopharmaceuticals or peptide or protein-based drugs are to improve the stability, physical stability, and this can be done by appropriate choice of the unnatural amino acids. For example, if the stability is not good, one can alter few certain amino acids, identify which amino acid that's responsible for instability, and that can be then modified. The whole API can be modified to with enhanced stability. And the other problem is that controlling the PK parameters. However, with the appropriate polymer particles or microspheres, one would be able to achieve the sustained delivery of the drugs. And unfortunately, all these APIs have to be administered through invasive manner. And however, there are some non-invasive administration routes are available. Again, this all depends on the property of the API. So, and in addition to that, one of the greatest problem associated with these large molecular, macromolecular drugs is that they can be recognized by macrophages. So, to avoid that, one would frequently use pegylation. The pegylation basically increases the make the surface of the protein more hydrated and eventually escape the macrophage attack. So essentially, that's one of the greatest advantage. We're going to see the advantage of pegylation in the subsequent slides. And then the protein or the API can be attached to a certain cell surface receptors eventually, so that to improve active transport of the API. So by attaching a site-specific targets, one should be able to overcome any kind of passive targeting. So this makes highly specific to the certain tissue. However, the one of the challenges that high dosage requirements, because as I said, the low volume in the subcutaneous injection is a major drawback. However, by having a formulation along with the excipients, one would be able to overcome this challenge by preparing a good controlled drug delivery system. There are several nanotechnologies available. Basically, these nano formulations have the ability to deliver the macromolecular drugs in a quite sustained manner. And there are two advantages of this nanotechnology. One is the enhanced solubility and dissolution. And then because the nanoparticles are larger in surface area to volume ratio, they offer a greater solubility and tissue penetration compared to the free active pharmaceutical ingredient. In addition to that, the nanoparticles, by having an appropriate size, and they can prolong the residence time through systemic circulation. So, and they can be, modify the drug distribution and may permit drug targeting and transport across the biological barriers. Now, once the drug is administered, it enters into the systemic circulation, and the drug has to pass through the endothelial lining in the vasculature. So in most part of the body, the endothelial lining is continuous. There is no any kind of a defect in their structures, which makes all these macromolecules highly impermeable. And in general, there are three kinds of endothelial structures are known. One is the continuous endothelial cells, where there is, and these cells have a very tight junctions and a continuous basement membrane, which prevent any absorption of the drug, and is often found in brain, lung, and muscles. The other group of endothelial cells are fenestrated endothelial cells, and where the gaps vary between 20 nanometer and 80 nanometer between them, and which would allow the passage of small molecules. However, such fenestrated endothelial linings are found in capillaries in the kidneys and gastrointestinal tract. And the third one is the sinusoidal endothelial cells, where the gaps are a little bit larger and go up to 150 nanometers. The basement membrane is either discontinuous, as in the capillaries of a spleen, or absence altogether. So additionally, the integrity of the endothelial barrier can be disturbed by inflammatory process or by tumor growth. So in the case of tumor, as is shown here, this creates a permeable vasculature, and the nanotechnology basically take advantage of that, and using this, the porous nature of the tissue, the tumor tissue, and one can develop a particle size of 200 to 300 nanometers that makes these nanoparticles permeable. In many of the success of the nano formulations for cancer treatment is basically due to the enhanced permeation and retention effect. And as I said, it's because of the highly permeable endothelial cells of the tumor sites. Now, here is the list of APIs that are useful and that have been commercially successful. And if you could look into that, many of them contain either polyethylene glycol or poly-DL lactates. They have in the form, and they can be administered either in the form of nanoparticles, liposomes, or polymer protein conjugates, or antibody polymer conjugates. So there are, the technology hasn't gone beyond PEG or lactic acid based system, and largely because they have numerous advantages over the other polymers. If one administers systemically, the renal clearance is dictated by the molecular size. Once the polymer can be conjugated to the active pharmaceutical ingredient, such as a proteins or peptide drug, and if the molecular weight is more than 45,000, they can overcome any kind of a renal clearance. So, and however, if the size is a little bit larger and is highly hydrated, and then they cannot be, identified by the macrophages present in the system. So, which makes the polymers quite unique in the delivery and of antibodies or large biopharmaceuticals. In particular, the PEG, as shown here, has a structure which is shown here. The presence of the ethylene oxide ring, which is shown here, can actually take up 4 water molecules. And these water molecules are present on the surface. And if you have an active ingredient inside a nanoparticle, and the surface is decorated with the polyethylene glycol, what happens is that in order for these two particles to coalesce, there's a lot of penalty. All these water molecules at the interface have to be removed, which is extremely difficult. So, which makes this individual, which makes, a better stability. So, therefore, the presence of PEG gives a better stability to the nanoparticles. And in addition to that, the PEG is being a hydrated polymer can enhance the solubility of highly, insoluble active pharmaceutical ingredient. And once since the polymer is on the outer surface, any proteases would not approach because it needed, a lot of penalty is needed to remove this water molecule from the interfaces and to really access the active pharmaceutical ingredients. And in addition to that, if the macrophages, which can capture the large molecular weight or any foreign bodies, would not recognize because the surface is essentially highly hydrated and never recognize those such nanoparticles. Therefore, the PEG is quite one of the major ingredient in any nanoformulations. Having said that, now let's go into some of the details, how the polymers structure can be manipulated to produce drugs of controlled drug delivery of pharmaceuticals. The first one is the polymeric micelles. And just like these micelles are like a kind of a soap solutions, you would have seen that, when the soap molecules is like a lollipop, a small, a stick-like hydrophobic chain, and the head group, which is a hydrophilic. As the concentration of these polymers are increased, they tend to assemble into a spherical form known as micelles, and the concentration which this transition happens is known as the critical micellar concentration. So once they form a core of hydrophobic, and then the shell is hydrophilic, which makes that unique pattern. So if we have an hydrophobic drug, they basically enter inside the hydrophobic region and basically prevent any kind of hydrolysis. That's one way, one could prevent the hydrolysis of any active pharmaceutical ingredient. In general, polyethylene glycol or a polylactate or a glycolic acid polymers are used for this purpose, and the molecular weight can be manipulated. The composition and molecular weight can be manipulated, and that can give greater stability. Right? So if you have low PEG, the polymer degradation time is low, and if you increase the block of polymer content, the lactide and glycolate content, then one would increase to 2 to 5 weeks. And if you use a different polymer, polyepsilon caprolactone, the degradation time increased to more than 12 months. So by incorporating the drug, one would be able to measure the high encapsulation efficiency, that is the amount of drug that enter into the interior of the micelles, and also one would be able to calculate the total content of the API. So there are some guidelines available to quantify the amount of API in the micellar system, and all this can be easily done in a typical pharmaceutical labs. The next one is the liposomes. Unlike micelles, liposomes are not like thermodynamically driven. Basically, they need energy to form a liposome structures like this, either in the form of sonication or vigorous shaking. As you know, these are lipid formed by the lipid molecules. They have the tail here, and then the head. They have a hydrophobic head. And depending on the method of preparation, one could prepare small unilamellar vesicles. And if the advantage, unlike micelles, the interior of the liposomes are hydrophilic in nature, highly hydrated. Therefore, for nucleic acid delivery or large macromolecular proteins that are highly soluble, they can be delivered by using the liposomal formulations. And either one could use a small unilamellar vesicles or large unilamellar vesicles or multi-layered vesicles. So all this can be useful, and you will see how the principle behind the COVID vaccination that saved many of our life. When it comes to subcutaneous delivery of the antibodies, there are more than 100 antibodies have been are available, and one way to deliver them is through having a biologic that are coated with the polymer particles, and these polymer particles contain so they're protected by hyaluronic acid. And because there are a lot of hyaluronidase present in the subcutaneous tissue, it will break down and expose the nanoparticles, which eventually deliver the drug molecules. Some of the polymer microspheres, particularly the PLGA-based microspheres, are quite useful. The greatest advantage of them is that they have the degradation rate can be easily, manipulated by varying the ratio of lactate to glycoside, and also the molecular weight also can be, quite varied depending on the method of synthesis. And also, they are quite usually there are a lot of manufacturers who could we can produce, PLGA particles in large quantities. So as I said, if it's acid terminated by appropriate choice of the L by lactate versus glycolate ratio, one would be able to have manipulate the degradation time of these nanoparticles of the microspheres. And here is an example that uses the microspheres for control for the subcutaneous. This is again one of the successful commercial formulation. I want you to watch this video.
Eligard, leuprolide acetate for injectable suspension, consists of an active drug, leuprolide acetate, in the Atrigel delivery system, an innovative dual liquid polymer. Uniformly mixed biodegradable copolymers with even distribution of the active drug suspended within the carrier enables a consistent release of leuprolide acetate over time through an in situ delivered implant. A short safety needle is inserted through the skin at a 90-degree angle into the subcutaneous space. There are many sites with adequate subcutaneous tissue, and administration is simple and straightforward. Eligard is injected as a liquid, and it becomes a small pool as the needle is withdrawn. An exchange of the biocompatible liquid carrier within the Atrigel delivery system and the subcutaneous aqueous fluids causes the polymer to rapidly solidify, capturing leuprolide acetate molecules in a solid, stable, biodegradable implant. The Atrigel delivery system provides extended controlled drug delivery through optimized copolymer ratios that enable consistent degradation throughout the dosing period. 1, 3, 4, and 6-month formulations are available. Throughout the dosing period, Eligard slowly degrades, releasing leuprolide acetate into the subcutaneous tissue. As the Atrigel delivery system's biodegradable polymers hydrolyze into lactic and glycolic acids, leuprolide acetate is released and enters the subcutaneous space, then enters the bloodstream. The acids are safely degraded into the byproducts carbon dioxide and water. Continuous and sustained drug release over a targeted time period results in persistence of drug at active concentrations throughout the entire dosing period. The active drug leuprolide acetate travels through the circulatory system until it reaches the pituitary gland, where its mechanism of action inhibits the release of luteinizing hormone normally produced by gonadotropic cells in the anterior pituitary gland. Luteinizing hormones act on Leydig cells in the testes to produce testosterone. Hence, Eligard prevents testosterone synthesis in the testes. Prostate cancer is the second most common cancer in men in the U.S. and is a testosterone-sensitive tumor. Suppressing testosterone production with Eligard can prevent the stimulation of prostate cancer cells to grow. Eligard is a GnRH agonist indicated for the palliative treatment of advanced prostate cancer. The Atrogel delivery system delivers consistent, controlled, extended release of leuprolide acetate to suppress testosterone and inhibit the growth of prostate cancer cells.
So basically, if you could look into that, it has multiple, principles are involved in this. Here, you have the biodegradable polymers and then leuprolide acetate, which is a peptide-based hormone, are used. And once they form a microspheres, they are now added a solvent, which is known as N-methylpyrrolidone. And interestingly, N-methylpyrrolidone has excellent safety features. It doesn't have any adverse effect to human localized tissue, both systemically as well as to the localized tissue. And since the subcutaneous space is highly hydrated, there's an exchange of the solvent which occurs very slowly. And basically, when the injectable is injected to the subcutaneous tissue, they remain as a solid mass. And eventually, it doesn't need actually any kind of an implant to deliver. It's basically the solid mass formed by this principle would basically deliver the drug for a longer period of time. So that's some of the advantage of using this such system. There are quite a bit of marketed PLDA-based microsphere-based drug delivery systems are here. And what I want you to do, I want you to go through them. And some of the principles are basically same. And the PLGA has the greatest advantage that after degradation, they produce carbon dioxide and water, which does not have any effect to the human tissue. Now, having said that, when we have we have talked about the protein or peptide-based drugs, and what about the nucleic acid therapies? And usually, when it comes, nucleic acids are unlike proteins. Nucleic acids are formed by the phosphate bonds, and these are negatively charged, polymers, and the molecular weight also quite vary. And either they can be inject, the therapeutic strategy is to use antisense or a small interfering RNA or aptamers or as an mRNA. So depending on the kind of strategy one would be able to develop as an excellent drug delivery platform. So in general, the challenges associated with the nucleic acid therapeutics are they are easily susceptible. The phosphate bond can easily degraded by the nucleases present in the bloodstream. And because of the large size, they have very poor penetration and can be readily eliminated by human immune macrophages. So therefore, they have to be administered through systemic routes only. The nasal or pulmonary routes are also not useful because of poor systemic absorption, and these are being negatively charged. They can be easily attached to any of the, neutral or positively charged surfaces in the body tissue. And again, the oral route is impossible because they can be readily degraded under acidic conditions. So in general, there are two different routes. One is the viral delivery systems, which have been coming up in a big way, and the non-viral delivery system, which uses cationic lipids, polymers, and peptides, and which are useful in the delivery. Usually, when you have a negatively charged nucleic acid, and then when you add cationic polymers or cationic lipids, they tend to form a complex known as polyplexes, and this polyplex basically shrink the whole structure of the DNA, preventing the exposure of the DNA to the nucleases. Excuse me. So in general, when during the formulation strategy, an excess of polymer is added so that, to stabilize the nucleic acids against nuclease degradation. Kind of wide variety of polymers are available, but the most successful one commercially available is the polyethylene amines, and it has a high transfection efficiency over other polymers, and we will see how they helpful in delivering the drug. Now, here you prepare a liposome that contain branched polyethylene amine, and then the mRNA is inside because it's being an hydrophilic in nature, it's inside the liposomes. So after injection, and once the drugs are delivered to systemically, it gets into the bloodstream, and you know very well, because of the APR effect, they get absorbed into the tumor sites quite, because of the permeation, high degree of permeation caused by the tumor tissue. In addition to that, these polyplexus particles are more than 6 nanometers in diameter, so therefore, which prevent any clearance by the kidneys. At the same time, because once you decorate them with a polyethylene glycol, one would prevent the macrophage capture. So by appropriate choice of the polymer, one would be able to obtain the control and successful delivery of the nucleic acid particles. Once they are inside the cell, they are being taken up by the cellular systems. And since you have a cationic amine group, these amine groups will now, and inside the compartments, it's acidic in nature. They becomes eventually cationic positively charged. And this charge drive the chloride ions from the further extracellular, the inside the cells, the surrounding cellular environment. And as a result, what happens is that there is a significant osmotic stress developed inside the cells. So as a result, what happens is that the cell, basically the compartment breaks down, and eventually the release of the nucleic acid. So therefore, in any of the nucleic acid delivery, if you can see, it has four important components: an ionizable lipid, meaning that essentially it's ionizable means a cationic lipid, so that to give an osmotic environment, and also the helper lipid that resembles, very close to the cellular membrane, so that it can be taken up by the resident cells. Also, they all do contain cholesterol to stabilize the lipid layer and also maintain the fluidity of this structure. And finally, PEG to improve the stability and reduce the protein absorption. Sorry. And these are some of the successful, strategies using the liposomal formulations, and then you can go through some of them. Essentially, the principle is very similar. And I think most of us have vaccinated with the Pfizer BioNTech vaccines, right? Some of the components of them are shown here. The structures are shown here: ALC 3015, ALC 0159, and DPSC, and cholesterol. So what I want you to do is that I want you to take a few minutes, 2 or 3 minutes, to answer this question. And the ingredients are given, the structures are given in the previous slide, and then find out the role of individual components. I'll give you 2 minutes of time. Okay, let's reveal the answers. mRNA is actually the active pharmaceutical ingredient. ALC3015, if you look into that, it's a cationic, the structure has a cationic group, like a branched ethylenimine kind of a group. So it's a cationic lipid. And then ALC0159 is basically a helper lipid that, it has a um, polyethylene glycol unit, and which is useful in preventing the macrophage capture and also renal clearance. And DPSC is basically a helper lipid which enhances the cellular uptake by the host cells. And the cholesterol is basically for structural integrity. I think, and that's my last slide. And I thank you very much for your attention. And and feel free to contact me if you have any questions on any of the topics that I covered in the last two lectures. So, and thank you very much. And I would like to answer if you have any questions on. Thank you, Professor Sriram, and thank you all of you.
Yeah. Guys, any questions for him? No questions, right? Let me check the online. Any questions for you? Online. I think. No, online also looks good.
Okay.
Let me stop that coming.